and welcome to this seminar, which is the first one in this year. It's a pleasure for me to introduce our speaker today, Mikel Valle. He has focused his activity on structural biology, specifically in cryo-electron microscopy and 3D data processing. He started in the field of electron microscopy during his PhD thesis period in the group of Professor Carrascosa at the CNB, at the SIC. And later he joined the group of Professor Frank in Albany as a research associate. The group was a leader in the application and development of cryo microscopy and in the study of the structure and function of ribosomes. During this period, he published several seminal works on the structural basis of translation in important journals as Science, Nature, and Cell. And important to say that Professor Frank was awarded the Nobel Prize of Chemistry in 2017. In 2006, Mikel Valle moved to CIC BioGune, and in this period, he has established the foundation of a new cryo-EM hub. And in all this time, this facility has grown in the number of research groups, and the weight of this community has been recognized with the acquisition of a high-resolution electron microscope that is being installed at the moment. He has established and developed several research lines focused on different biological samples, and the most important line of research in his group has been focused on the structure of flexible filamentous plant viruses. This line started 8 years ago with collaborations with different groups, in particular Dr. Aranda at CSIC in Murcia, and later with Dr. Pons at INE, which established a Spanish network of groups that brought together the fields of structural biology and plant pathology. His research has delivered the first cryo-EM structures at high resolution for several representatives of this group of viruses, and they have also unveiled the structural homology between nucleoproteins of previously unrelated single-strain RNA viruses. All these previous studies about flexible filamentous plant viruses have allowed to detect a previously hidden and universally conserved single-strand RNA binding pocket in the viral nucleoproteins. So, as I said, it's a pleasure to have you here today, so thank you for accepting our invitation, and now you can start with the seminar.
Okay, good morning, everyone. Thank you for the invitation to come here to present our work. And it's a real honor to be here, one of the main pillars of the structural biology in Spain. This is more or less the distribution of our center or institution is in the Biscay Technology Park. We are distributed in 3 different buildings. The mother building, which is more or less dedicated to cell biology stuff. The building 800 where we are is the hub for structural biology. We have X-ray crystallography, NMR, and electron microscopy, and we have another additional group here in this another building with a special setting as for instance, a P3 to work with prions. So this is the distribution that we have. And we have a novelty that is not in our building, it's in the biophysics unit, but it's just installed. It's a Titan Cryos G4, which is the best equipment you can use nowadays for cryo-electron microscopy, and delivers very high resolution in routine work. So this is going to change our life, definitely, because in the last, probably everyone is aware here that cryo-EM has now an explosion in the last 7 years or so, and it has been kind of difficult to cope with this, to deliver high-resolution structure when you don't have a high-resolution electron microscope around. We have lagging times 3-4 months, for instance, to get access to one facility, and the last 2 years It has been really crazy. So this is going to change our lives, and it's going to change the way we face some project, because we have no access, and we can analyze, instead of one sample, we can analyze 4 or 5 different samples. So we hope this is going to change our life in the near future. Actually, we have the scientific opening of this microscope this Friday. It's ready now, which is very good. It's all the way around. Okay. So I'm going to present one of the main lines that we have is regard those filaments that we see here in the cryo-EM image. I'm going to start with this painting, which is a funny one. This painting is from the 7th century from the Netherlands. It's a very famous one. It's anonymous. And it depicts what is a variety of tulips, which is, I think, Semper Augusta, something like that. Probably you know that in that time in the Netherlands, the Dutch people, they went crazy about tulips. And they got the first economic bubble that has been described. And this is the most expensive tulip they got at that time. And it was expensive because this kind of crazy pattern of coloring, the white stripes on a red background, and because it was really difficult to multiplicate the plant to get new bulbs. And the two characteristics are because it was infected by a Potyvirus, one of those viruses. So it's the first contribution described for these viruses to have economic impact. But they have a huge economic impact in economy nowadays in agriculture, as you may imagine, because they infect plants. So they are essentially, as I point here, they are single-strand RNA positive polarity. Monopartite is just the entire genome in one molecule. And they are protected by nucleoproteins, hundreds of copies of the same nucleoprotein, are arranged in helical fashion. As you may see here, they are long, they are thin, and they are flexible. Those nucleoproteins in the field, they were called code proteins. But you can call it, you can name it the way you want. So, they are, as I mentioned, they have a large impact in agriculture. There are more than 400 different species of viruses. They are distributed in different families. The largest one is Potyviridae, where the Potyviruses are. They are in the same group, in general group of flexible filamentous plant viruses, because the virions, they look very similar. But for the rest of the characteristic, they are different in terms of structure of the genome. If you compare their RNA-dependent RNA polymerases at the sequence levels, they are distant. If you try to align the sequence of the nucleoproteins, you cannot get a match. You don't get an homology at the level of the sequence. So, and those are negative staining images that were available at the time. Examples, I can go through a lot of different diseases that they can produce in agriculture, but I just want to highlight some of them. For instance, a combination of 2 viruses from different families in sweet potato. This is relevant in Africa because they have outbreaks of the viruses, and it's really a disaster from the humanitarian point of view, because sweet potato is the main source of calories in some countries. We have in potato, the most important viral disease is caused by potato virus Y, it gives the name for the Potyvirus group. And potato virus X, this is a problem. Plumepox virus and citrus, they infect trees, which is a disaster because they kill the trees. And in some instance, for instance, in Europe, in glass houses in tomato, pepino mosaic virus is always horrible. They produce this marveling, and you cannot market the fruits. And watermelon mosaic virus in the Mediterranean basin at the level of cucurbits. So, as I mentioned, they have a huge impact in agriculture, but they are also used in nanotechnology and biotechnology because they are, polymers somehow. In nanotechnology, you use polymers, and they are usually being tested as vaccines because it looks like you have having kind of the same epitope. You can modify genetically the nucleoprotein, and you expose one epitope in all the copies, and it seems that have this kind of repetition is a good way to present the antigen to the immune system. So there are several applications you can do, and in the time, for instance, modifying the nucleoprotein in the virus to expose something was made blindly because the structure wasn't known. So it was try and error, try and error, try and error. So at some point we contact with the group of Mikel Aranda, and we start thinking about the structure of the virus. At that time, what was available was crystal structure of a fragment of isolated nucleoprotein that was expressed in E. coli. I think I've broken this thing. Okay. And several cryo-EM maps at intermediate resolution, 15, 20 amps. Actually, they were depicting the helical arrangement as a right-handed helix, similar to as rigid viruses as TMB, for instance. Sorry. There you are. So the idea is what the structure, the arrangement of the virus was not known. So we decided to start working with those viruses. As I mentioned, we have the development in cryo-electron microscopy, and in order to analyze helical symmetry samples, you can go in the Fourier space using Fourier Bessel indexing or helices, or you can go, in this case with the high resolution cryo-EM, you can go to single particle, so to speak. The problem with Fourier Bessel is that you need to have a lot of asymmetric units in register, and which is something that is flexible. This is not possible. So you never reach high resolution. But here, what we do is we select stiff segments, straight segments of those filaments, and then we chop them in different segments with different overlapping. This is something that you can play with, and it's not very relevant at this point. And then work with them as single particles. In the single particle approach we use usually in cryo-EM. And taking advantage, of course, of the helical symmetry. Helical symmetry is taken into account during the alignment, during the reconstruction, in all the steps. And it multiplies the number of, at the end you get a lot of asymmetric units that contribute to the final average you're going to calculate in cryo-EM. So we start for the first sample, because Mikel Aranda, working in plant pathology in Murcia, they were very, this one was one of their favorite viruses. There was a problem in tomato culture in Murcia. And essentially it belongs to one of the families, Alphaflexiviridae. And despite was discovering one pepino, actually the name is come from Spanish, from a sweet cucumber from Peru, it was spread all over Europe and North America very rapidly. Well, we don't know whether the virus was there, but detection was new, and it was tested. It gives you a lot of different problems. Most of the viruses they affect color with mosaics, for instance, shape, size of plants at the level of leaves. Oh, this is a nightmare. And fruits at the level of coloring, size, yield. And for instance, those tomatoes, you cannot use them in the market. You can use it for tomato sauce, whatever. And the price goes really, really down. So, and with the margin they have, it's just devastating. So as I mentioned, what we do is collect images in different microscopes. I think that this one, this was in the MRC, because they opened a line for a Spanish researcher. And well, the work includes some kind of testing different parameters in the helical symmetry. You have to figure out the rise, the rotation, all the pitch, and the number of units per ton. And as I said, using the single particle approach and taking advantage of the helical symmetry, we got a map about 4 Armstrong resolution. The average in our case is always more or less the same. It's the data set the size of the data set we can collect, and the flexibility, it imposes some kind of restriction in the resolution. And in most of the maps we have worked with, we always get about 4 Armstrong resolution. And we can see here, for instance, estimation of the local resolution. The red one are the regions with poor resolution. So what we learn about the structure, I think this is kind of crazy coloring, but it's just to see the light blue is which is a core domain, and we have arms, the end flexible arm that is bridged into subunit in purple, and in the inner side we have the C-terminal region. The red displays the RNA in different terms. This is a cutaway to see the inner side, and this is just the surface. We always have a missing stretch of residues in the N-terminal domain. This is the most variable part in all those viruses. And this is just floating around in the outer sides of the helix, and in the three-dimensional average that we calculate, they are vanished because they are flexible. This is the one subunit, one isolated subunit, and we have here the signal for the RNA. What we learned there, first, as you may imagine, interaction. The N-terminal, in this case, for this Potex virus, family Alphaflexiviridae, we have a stretch of relatively aerophobic residues that go to hydrophobic groove in the next subunit. Is the reason for polymerization in one turn. For the inner side of the viruses, this is a network of very complicated interactions that implicate C-terminal domains at different levels that are in the inner side of the virus, and they establish a huge network of interactions. I'm not going to go into more details, but the RNA, and here we start with problems, because as we calculate an average, a three-dimensional average, the composition of the nucleoprotein is always the same, because this is the same composition, this is the same protein. However, the RNA is not. In each segment, you have a different sequence, the corresponding one. And we always have, as you can see here, the red region of the density for the RNAs, indicating that you have problems of resolution and definition there. And actually, in the first map, we could not even describe or define polarity of the RNA. We could not see even where the bases were pointing to. So everybody in the field present yet a poly-U as a model for the single-strand RNA, and that's what we did. But the few things that we could learn is that we have a stretch of 5 nucleotides per subunit, which fits really, really well with the length and the number of CPs. And also that no matter what the real orientation and the structure of the RNA was, we always had this kind of one nucleotide sticking out from the rest. It was pointing out some kind of RNA binding pocket at some point. So this was Alphaflexiviridae family. We went then to Potyviridae family. As I mentioned, there is no sequence homology between the nucleoproteins. So the approach is similar. In this case, it infects cucurbits in the Mediterranean basin. In this case, we use different software, for instance, but more or less the results are very similar. Different microscope also, but we end up in similar resolution. Not alone a large number of segments at the end, or not a large number of asymmetric subunit, but enough to describe, to put the protein on register and to describe in this case, a Potyvirus. What we obtain, the helical arrangement is almost identical. They have the similar arrangement. They are also a left-handed helix. And novelties here is that the N-terminal region that in the previous case was side-by-side interaction between subunit. Here there's a sharp turn here and goes to make an interaction with the subunit that are in the next turn on the helix. So it's a dual role within the same turn and between turns. The interaction between subunits in the same term is hydrophobic also. The other one is electrostatic with the acceptor here, the acceptor region in red and the donor region in blue. They are complementary. But the interesting point comes later. That you can superimpose both structures very clearly. They are the same fold, exactly the same. They are different most of the difference comes from the C-terminal part that is inside the virus. So, despite not having sequence homology, there's a clear structural homology. Okay, but this is really usual. It's not a novelty at all. But however, when we start using the same fold to pick for homologous using DALI server and this type of algorithms, we picked something that was really striking. It was that they had very similar fold with nucleoproteins of other type of viruses with segmented RNA with negative polarity envelope, and viruses that infect in most of the cases animals instead of plants. The structural homology between viruses at the level of capsid proteins in causal viruses is really assumed nowadays. But there was a huge discussion in the field whether those viruses that are not icosahedral, what can we use to make this type of classification based in the structure? And it seems to me that it's just the nucleoproteins, those that bind to the nucleotide directly. And you can see the settings. The structural homology includes even flu virus, not entire nucleoprotein. I think this is the N-terminal half of this protein. Or Rift Valley fever virus, Bungia virus are most of the viruses that we cut. And you can see the nucleoproteins here are inside our envelope, and they don't form helix, maybe helix-like structure at some point. So, for instance, we can pick two, Pepinomosa virus, the first virus that we described, and this one from Rift Valley fever virus, a Bungia virus, and they have an terminal here and alpha helical fold, but they don't look really similar. But if we just remove whatever is not the same, or whatever we cannot align, what we have is a very similar setting with the N-terminal in the same place, with the RNA group in the same place, C-terminal in the same place, and topology is the same here. So, which are the implications here in terms of building up a nucleoprotein complex? The different parts are in gray here. In Bungia virus, the C-terminal part falls around the main domain and extends the RNA bind inside by 2 nucleotides. Instead of having 5, we have 7. Here, the C-terminal domain contributes to the axial oligomerization to build up the helix. So, at the same time, those oligomerize also by the N-terminal region, and they use the same acceptor region, the group here. So everything is relative. The relative position is exactly the same. That's why, in one case, for flexible filamentous viruses, we have building of helices, interactions with the C-terminal region, and here we have kind of change of beats that sometimes look like they are making some sort of helix, but they are really flexible, so they can pack inside an sphere protected by the membrane. Actually, those in crystallography, they have seen tetramers, pentamers, and hexamers always sorry, always making circles. Okay. So our interpretation of if you try to compare nucleoprotein from one of those groups to other nucleoproteins of different families, you don't get a match in terms of structural homology. This only has been captured using one of those flexible plant viruses, the nucleoprotein for that, and with one you capture some of the representative here, and with the one some others. So our interpretation is this is a fold in a common ancestor, a nucleoprotein fold that has been transmitted. You can imagine that, evolution of viruses is just crazy. You have horizontal transfer and whatever all over the place. But here what we have is a naked structure that has to protect the genome in a very strict way. So our interpretation is that restriction in evolution has caused that the change, that this nucleoprotein has not changed very much. It's very similar to the common ancestor. And here, nucleoproteins that are protected inside an envelope, they have been capable to change more dramatically than those ones. So evolution here is divergent, a lot of different nucleoprotein settings. Probably they adapt to the host in order to replicate inside the cell host, so they can follow different strategies. Even then the nucleoproteins look different. But here we need to have a filament protecting the genome. So they don't have a lot of margin to change their structure and explore different other possibilities. Other stuff we have been working, for instance, we did work with turning mosaic virus. This is another Potyvirus. One of the main reasons was to see whether this signature, the presence of the N-terminal region that breaches within the turn and between turns, it was a signature of Potyvirus, and it turns out to be the case. The Potyviruses, they have this double staple. And we analyzed also virus-like particles. There were transient expression of the nucleoprotein in plants, and you can purify filaments at different length, of course, because you don't have the genome inside. And what we see, we try to process then you following helical symmetry was a disaster all the time. So without symmetry, you can see here that this is a classification, and there is no clear definition. So the absence of the RNA promote that you really are losing some, a good interaction between the proteins and the final filament. Even if it looks nice, a low resolution, if you try to define it a high resolution, you get nothing. Well, in this case, it turns out that it helps us to understand the role of the RNA. This is the atomic model of the viruses and not the BLP. And you can see here that it's a network in the boundary between subunit. And what is nice here is that both the N-terminal region, which is this one, and the acceptor region, which is this one, the acceptor for the N-terminal, or the other subunit, both of them depend of the interactions between some residues with the RNA, particularly those arginines here, and some residues with the RNA from the N-terminal arm. And between them, also, you can see the detail here. For instance, this arginine that has goes kind of a tertiary interaction with RNA and with the subunit that is in the neighborhood. So the point is that missing the interaction with RNA, they don't lock properly both the N-terminal arm that is going to breach between subunit and the acceptor region in the neighbor subunit. You need both of them. This is just a curiosity because we started this. The idea is, of course, this is the average of the filament, but what happened in the ends of the virus is how it's capped. Nobody really knows that. And so we decide to, okay, let's see whether we can, because in 2D, you want to miss a lot of different projections. So you need to have tilting somehow. For that, we faced tomography. And we never did tomography before. And we started this in February 2020. It was a disaster. We collected the data in February 2020. And because we scheduled a workshop in tomography in March. So everything was canceled, of course. And we have spent 1 year and a half trying to process this data with no help with nobody. But the idea is, in this case, for Potyvirus, which is really relevant, Potyviruses, as I mentioned, they are all the outsiders here. They have a protein linked, BPG linked to the 5' end, a poly A tail I didn't put here, in the 3', and they code for a polyprotein that is processed later. They are more like in the picornavirus-like group of viruses, because of this structure of the genome, or the strategy they follow, the presence of the protein. And we choose that because if it has a protein in one side, maybe we are lucky and we can see something there. So the idea is that you make tomography, make a tilt series, you align them. This is one of the images of the tilt series. So the idea is to select the ends of those viruses. When we calculate cystotomogram, this is just different sections at the end of the tomogram. You can see that the gold particles that we were using for alignment, which are fiduciary marks, are removed before going to the tomogram. So we have three-dimensional data here, and we can select the ends of the viruses and then make single particle averaging technique, but instead of using two-dimensional images and go to a three-dimensional final result, we have three-dimensional objects from the beginning, and we can align them. The problem, you may imagine, you have those are 100. This is a very good picture of a tilt series, because we have a lot of ends here, but this is not usually the case. So with I think it was 80 tomograms or something like that, we collect around 600 ends, which is really, really a poor number. But it was just kind of proof of principle to see whether we could get something. So the final average we got is this one. We always had around 30 Armstrong, which is really bad if you want to go really into details. But at least we have evaluated the technique, we have evaluated the type of result we have, and whether we can, we collect those images in a 200 microscope, which is even worse. So we know what to do in the next step. Well, the helix more or less can be, you can more or less guess what it is, even sometimes a globular region, one nucleoprotein, so to speak. And we did several trials, different type of processing, and what the only result that came out several times was this one, more or less 50%. You have one or the other. Of course, we did classification using more classes because you may have broken filaments and you are not aware. At the same time, I have to say that we prepare those samples for tomography the same day of the purification. When just prepare viruses without storing them in any place, and we make the grids. So trying to improve the number of real ends in the sample. And when you do classification, it is something. It doesn't look like there is nothing capping. It's like the helix has a stop at some point, and period. And we have focus classification here in the tip with the two classes, kind of a picking end, some crazy stuff here, more thin helical something, and analyzing them later. 30 answers, so we are not sure. But the only thing that fits in what we know, because we know the polarity, is that this could be the BPG that is linked to the genome, because the shape fits. This is some flexible region. This is an ensemble from NMR. The red residue that is here is the one that is attached directly to the RNA. So in the distance and orientation, it fits. So we think that this is the protein, but we don't know for sure. And this crazy thing, the only thing that we can think of is that the C-terminal regions we know that they reach higher levels in the term, they are exposed directly to the solvent. We don't know whether this is real or not. So we need to go to add more data here. So conservation, those are Alphaflexiviridae family, a Potex virus, and Potyviridae family, Potyvirus, and they have the same fault. What happened with the rest of them? Well, this is a more detail. This is different terms of the you see the electropositive region is in the groove, in the floor for this turn, and in the ceiling from the other turn. So it's a good place to put the ribonucleic acid there. And as I mentioned, we didn't know exactly the position, but we always have, this is the core domain, this is the lone helix, and we have one one base just pointing here to a binding pocket. And we start watching residues that are interacting with the RNA, the analysis, the first analysis. And then we realized that despite not having sequence homology, there were some residues that were in the same place in both viruses. You know, the serine here, well, there are two here. Aspartic arginine, lysine here. So we start wondering whether there was something special there and there was some kind of conservation. Also, there is the aspartic acid that is conserved. It has always kind of a signature with hydrophobic residues that make the floor of the RNA binding pocket. So more or less they are organizing everything to put the aspartic acid in the proper orientation there. So what we did, as I said, when you align by sequence the nucleoprotein of different families, you don't get anything. But what we did was calculate the consensus sequence within each family and then compare them. And you can see that there are 3 residues that are really conserved. And even the signature here of the hydrophobic residues around, they change, but they are also present. So of course, the idea is this RNA binding pocket is conserved, and probably the other 2 families that we have not worked with, Betaflexiviridae and Closteroviridae, they display exactly the same fold for the nucleoprotein. Very recently, the structural another Potex virus came out. And in this case, they start with a huge data set, and they reach 2.2 after resolution, which is really, really good. It is a very good work. And you can see the numbering is different because they display. So essentially, they can define perfectly how 4 nucleotides are following a canonical A form for the RNA, and the outsider is this one residue that goes to the RNA binding pocket, and it depends of interactions, hydrogen bonding between the conserved serine, aspartic, and arginine. Those 3 are conserved because they do something special. They put in this nucleotide in a binding pocket. Probably is the main engagement of the nucleoprotein with the RNA. Why are so conserved? No idea. No, we have no idea. Actually, I always say that they are universally conserved, but there are 2 outsiders here in the arginine, 2 out of 400, that they have changed for the arginine for a histidine. So it's not such a big change. So actually, when we described the first virus, Pepino virus, we didn't have any idea about the conservation of anything. However, it was very clear from the interactions that we were observing that there was some residue that may have some kind of impact. So we did test with Mikel Aranda. This is a transcomplementation assay. In one vector, you insert in the plant all the genes of the virus but not the nucleoprotein, not the code protein. This is substituted by gene fluorescent protein. In another vector, you provide the nucleoprotein. So if they complement each other, you have virus production inside, and you can follow the virus by following fluorescence. So you can check mutants and mutations in the conserved arginine, aspartic, even in this lysine that is conserved between some Potyviridae and some Potex virus. What we see is that there is no signal for the presence of virus of movement of the virus between cells within the leaf. And for instance, here this glutamine that could be important if you mutate it, the virus is still alive. Actually, you can try to purify viruses, and we only get some viruses for this glutamine and the wild type. So this interaction is really important, and the level of interaction with the RNA and the movement of the virus inside the plant. And viral production. So functionally they are relevant. And what level, I don't know. So the idea is, okay, this is not an enzyme, which is, I mean, they are more than 400 different nucleoproteins that interact with RNA in a non-sequence specific manner. So in theory, there is nothing that would say some function that has to be highly conserved. So to be so restrictive from an evolutionary point of view, but they are very restrictive. So the idea is, can we take advantage of that? So we thought that maybe we could do some kind of drug discovery, develop, try to find some antiviral. It may sound crazy. Well, this is because also our institution is kind of forced to go to more applied science. So we decided to try. We were clueless about drug discovery, and we made a lot of different mistakes. So, but it's not that we are going to provide a phytosanitary compound to be used in the field. This is nothing, probably, not even socially acceptable. I mean, they are used, but to promote this kind of stuff is kind of hard for now, even to get funding. So what Miguel guided us to an application which is plant in vitro culture. In vitro culture can be named plant, tissue, in vitro culture, whatever. This is a technique that is used to multiplicate very rapidly, clone one plant of interest. And may sound crazy, but there are millions of plants just in Spain that are produced that way for agriculture. The idea is you want something that is very special, one characteristic, a plant that is resistant for whatever disease, and you want to multiplicate very rapidly. Instead of going through the seed production, the complete cycle of production of a plant, you can take a fragment of the plant, and from each fragment you produce an entire plant. You have just to put it in a culture media with some hormones, and you can reproduce the plant. And there are dozens of companies in Spain working in this kind of field. So the idea is that since the control, I'm not going to explain everything here, but controlling of viral infection is really difficult. You cannot do just prevention. And what works really nice is you So I was saying that we moved to assays in vitro culture, trying to remove those experiments that we start with a plant of tomato that are infected with the virus. We start with, I don't know, 90 plants to go in different sets, different controls to have significant from a statistical point of view. They run about 6-7 months. And we start those in 2020. So we have not finished any single experiment at any time. But we have learned something. I mean, we could not present the numbers because we have to stop and keep. It has been a disaster. So the idea, we learned something. Essentially, the high toxicity of this, you see, the plants have several concentration and they don't grow at all. And we have problems of solubility of those drugs in the in vitro culture media. So what we learned that we made a huge mistake, that we started with initial library compounds that are dedicated to drug discovery use the strategy of lead fragments for human use. And for humans, you need to have some kind of hydrophobic, more hydrophobic characteristic to cross the gut barrier and to reach blood. And this is not good for plants and for soluble media. So what we are doing right now, this is a collaboration with Gonzalo Jimenez Assess, is we start, instead of a limited library compounds, we are using in silico libraries, virtual libraries, with several characteristics, the partition number moving to the hydrophilic side with a limitation of compounds, and we have a set of, we use a thin library of commercial available in theory compounds. In the set that we have selected about 25, I think, we have protected 16 of them, and compounds that we are testing right now. This is just kind of a note at the end. Of course, the problem here is that it's not a regular drug discovery because you don't have one target. We have 400 different targets. And this is tricky because you can go to something that is general, that can be used with a lot of different viruses, or something that is very specific because it's particular interest economically or whatever. You can go to Potyvirus or you can go to Pathovirus Y. So here the idea that the structure information is required to see which are the main interactions that happen in between the compound and the nucleoprotein. And for that we did, as I mentioned before, crystallography. Well, it was a disaster. No, we didn't get anything. And now we are evaluating the use of viral-like particles. Those are viral particles of Pathovirus Y. They are really different from the ones that we analyze. They are expressed in E. coli, and you can see here they don't have a real canonical helical arrangement. They have stacked rings of each subunit, and they are rather, the only interaction is just the N-terminal that crosses between turns. And here we have evaluated that despite selecting a straight segment in the micrograph, we still have tilting and relative rotation. We are here at 5 Armstrong resolution or something like that, but this is the evaluation that this can be used in order to describe the interaction of the nucleoprotein with the compound. So we know that we have to scale up and start using a larger number of initial segments in order to get a resolution to describe the interaction. And that's what we are planning to do for the future. So I'm just done. So we deliver the structure of Potex virus and Potyviruses. This describes obviously the homology between them. There is also an homology with distant nucleoproteins of viruses that were not related previously. And within this conservation, inflexible filament of plant viruses, we have detected this highly conserved RNA binding pocket. Why is it conserved, we don't know, but we can play with it. And then we're trying to move to apply science with in the field where we have learned things. We have not been very successful right now. And A lot of people have participated here, of course. PhD thesis by Miguel and Rebecca, and work by Xavier with the first viruses. We have collaboration in NMR with the group of Oscar Millet, in flexible and rigid docking, virtual docking with Gonzalo Jimenez Osen. David Hill was in the electron microscope at that time, and collaborations with groups in plant pathology as a group for Miguel Aranda and Fernando Pons in different places. And of course, we collect data for free in the MRC at some point. As I mentioned, they open very gratefully, they open a line for a Spanish researcher, and we collect a lot of data in the Nanoscopy Center of the Netherlands, where the story starts with the tulips. So we are in the same place. And the funding. So thank you very much for your attention, and I'm willing to take questions right now.
Okay, thank you for the presentation, and we are sorry for the inconveniences with the audio. The people in YouTube missed some part of the final slides of the presentation. Now we have time for questions here, or also if anyone has questions through streaming, just write them in the YouTube chat, please. So questions here, comments?
Thank you, Michael. Mikel. I think it's a very clever approach that you are using for finding antiviral compounds. However, I wonder why are you targeting the RNA binding site rather than, for instance, these N-terminal or C-terminal ends that are building up the filament, because maybe this is the reason of toxicity in plants.
The main advantage of the RNA binding site is conserved along all the viruses. The N-terminal region is really divergent. It's crazy. You can, I mean, even the length, the differences in length in the N-terminal between some of the viruses is about 50 residues, longer than others. It is true that there is one special, some viruses they are, I didn't mention, I'm sorry, some are transmitted mechanically through wounds of plants, and they are in the environment and they get into plant cell, or they are transmitted by vectors, usually insect. And there is one particular region of those, a set of 3 residues, I don't remember exactly the sequence, that seems to be important in terms of, or required, not important, required, in terms of attached to the salivary gland of the insect and stay for a while and then being able to infect another plant. And this is one of the targets also in this type of things. But the problem there is that they are, the mechanism, I mean, the role of those 3 residues and interaction with the salivary gland of the insect is not known yet. So it's kind of a tricky field. I don't know about the about toxicity. What is the reason of toxicity? I'm more fond of that we have hydrophobic compounds and the plant they don't like at all. These kind of things sticking out everywhere. That's what I believe. I don't know, but I believe that this is the reason. But of course, I mean, the point is here, try to target something that is common to all of them, and then terminal is really, really variable.
There are questions here in the chat. One question from José Miguel Mancheño. How do plants fight against viral infections? In particular, is there any information about which viral proteins are recognized by the innate immunity system of the plant?
Immunity system of the plant. This is really difficult to talk about. Well, actually they use some kind of silencing RNA. But the person who discovered the silencing response of the RNA was Olivier Bonnet. He has retracted about half of his career. So I don't know what is true, what is not true, I don't know. I am clueless here. There has been a huge scandal in plant science in the last 5 years or so, with a lot of people making a lot of retractions regarding plant immunity. So I don't know. I don't want to go into that field because I don't know how to pick the good things from the bad things. With respect to those, for instance, with some experiments in plant, we have to introduce, not we, Mikel Aranda in the group, they introduce a protein that blocks the silencing RNA response in order to be able to express some vectors in the plant cell. In vivo, what happened? I don't know.
We have many comments today here. Luis Fernandez, thanks for your nice talk. A question: disorder seems crucial in both the N and C term segments of CPs. How does cryo-EM cope with this disorder?
Very badly. Okay. First, they don't have a defined secondary structure, so they are loops. They are flexible. I didn't even mention, didn't stop to highlight that, but of course this is the reason, because main of the surface contact between subunits are mediated by N and C terminal regions, so that's why the viruses are flexible. In our case, in our experience, the N terminal, we always miss whatever is not interacting with the neighbor subunit. The rest, this region is missed all the time. In some particular cases, it looks like, with some of the viruses, it looks like when the N-terminal stretch is really long in terms of amino acid, in the microscopy images, it looks like there are some small, tiny domains. So maybe there is a partial folding on some region, but it disappears in the cryo-EM map. We were thinking to use tomography also to pick those elements, but they are too small for tomography, so we cannot do that. In terms of C-terminal region, since it is really close packed inside the virions, for Pepinomosa virus, we can trace the entire C-terminal region and up to the L. With Potyviruses, in one of them we can trace the entire region, but in the other one we could not trace the entire, and we were missing the last 6 residues. That it looks like they were not following helical symmetry, but we don't know, because I mean, they were disorder in a cryo-EM that has followed helical symmetry. We don't think that they are going to follow a different symmetry. It's just because they are disordered, may have different options. And there's a difference because in Potex virus there is a central hole in the filament, and in Potyvirus is full packed. So they are, despite being or having the same fall and similar type of virions, they behave differently at the level of, for instance, VLPs. There are more examples of different VLPs, and sometimes you have something that is stacked rings, sometimes you have helical symmetry. In some cases, with some construct, they have an octamer. They make different crazy things, and it's difficult to anticipate which is going to be the result if you use one of those nucleoproteins and you remove some sections of the C or N-terminal regions. That's what we do, but it's trial and error. We cannot logically anticipate something.
More. Jose Miguel Mancheño also, do you see any evolutive advantage of filamentous versus non-filamentous virion-like viruses?
Okay, this is not a binary distribution. It's not 0, 1, but there is a clear trend. Plant viruses tend to be filament, and animal viruses tend to be envelope. There are filament viruses in one side and envelope viruses in the other side, but the trend is really, really clear. If you want to take the part of the membrane of the host cell by budding, you cannot do that with a filament because it's not possible. It has to be something spherical. So, and if you want to be an envelope virus, there are some topos around in one of the in these cytopos viruses they infect plant and they have a membrane. But the problem is that since you have the cell wall in plants in cell plant, budding is not forbidden, but is not favored. So membrane, taking the membrane from the host is a factor that prevents you to get a filament envelope virus, which it will be really expensive from an ethical point of view, and you may take half of the cell membrane of the host cell if you want to cover the whole filament with a membrane. Another point is that in plants, communication, the virus transmitted between plant cell through plasmodesmata. Those are small pores between cells, and they are narrow. And it's much better to have a filament that can cross between between cells rather than an spheric large structure that cannot cross, that cannot reach the neighbor cell. This is a discussion also because it is not known whether is the virion that crosses between plant cells or is kind of another type of nucleoprotein complex. What we know is that the interaction of the nucleoprotein with the RNA is strictly required to go to the next cell, because we have tested that with mutants in the RNA binding site, and we can see that if you mutate that, there is no the protein or the virus cannot be transmitted between cells. So the idea is that safe filament, they are allowed to go between plant cells, and the presence of the envelope and taking the envelope, the membrane from the host cell, that favors one morphology or the other.
We have more questions here, but maybe anyone here wants to ask something. Yes.
Have you considered testing different RNA polymers to see if you can stabilize the fragment, the protein fragment? Maybe you are lucky with some segment to see if you have better resolution.
The first thing is that we don't know, well, nobody knows, with how assembly and disassembly is regulated. We don't know. It looks like some viruses, they recognize a three-dimensional structure of one of the segments of the genome in order to recognize the RNA and start making the polymerization. Well, you mentioned to use different constructs of RNA to improve resolution. What we have tried is to define which is the determinant to oligomerization in order to get this short segment of RNA much shorter than the genome of the virus, and then made the tomography of the ends, because we can have short particles with both ends. But we couldn't get anything. No, it was not possible. We did different constructs of different RNAs and our collaborators also, but we never got any oligomerization in vitro.
Douglas Lawrence has two questions also. I was wondering if it is possible that the RNA binding site might also be conserved in some plant RNA binding proteins, and this could be the source of the toxicity.
That's a good one. But we did not find any structural yeah, okay. The RNA binding site. I never thought about that. Mm-hmm. Yeah, it could be. Yeah. It could be possible, yeah.
And another question also from Douglas is it possible to use the virtual screen to find compounds that bind to the viral protein but not the plant proteins? Thanks.
Okay, we can face that with AlphaFold too now. We can make models for in your plot system, whatever. Probably they're already done. I don't know. we could try that. Look for conservation of this RNA binding site. Yeah, we can do that. That's a good idea.
Any other question here? No? Then we thank you again, Mikel, for the presentation, and we also have a small present for you. So thank you.